Welcome to Ask and Learn.
Hello students, the question says that three gene angustifolia which causes angle, leaf vein, brevipedicellus which causes broadleaf, and the cut which causes cut leaf margin are linked on chromosome 4 of Arabidopsis thaliana. So these three genes are linked on the same chromosome in Arabidopsis thaliana. A normal appearing plant is taken from the wild and crossed to a true breeding triple mutant. So mutant which is represented as ABC is crossed with a normal appearing plant which is, maybe we can write it, normal appearing plant taken from the wild, and the mutant alleles are all recessive to wild type. So mutant alleles are recessive to the wild type, and order of genes is unknown. The following progeny phenotypes were observed, which are: the wild type are 2 numbers, angled, broad, cut or 4, broad or 400, angled, cut or 395, angled, broad 68, cut 64, broad cut 32, and angled 35. Now the question asked is, what is the genotype of the normal appearing plant, and what will be the correct map of these three genes on the chromosome? We know that the triple mutant is ABC, and all mutant alleles are recessive to the wild type, and we are giving the progeny phenotypes and their numbers here. And let's denote wild type alleles as A, B, and C. Since the normal appearing plant is wild type, it is wild type, so at least one of the alleles is dominant, or one of the alleles is wild type. However, we don't know if it is homozygous or heterozygous for each gene. So now let's analyze the progeny phenotypes to determine the genotype of the normal appearing plant. So since we have wild type which are 2 numbers, so 2 number of progeny is wild type, and this means that the normal appearing plant must be heterozygous for all 3 genes. Which is A, B, C, A, B, C. Now let's determine the correct map of these 3 genes on the chromosome. We can use the recombination frequencies between the genes to determine their order. The recombination frequency is calculated by the number of recombinant progeny divided by the total number of progeny. So recombination between A and B will be equal to, so recombination frequency of A is 68 and B is 35. So the recombination frequency between A and B, which is angled broad, is number of recombinants, angled broad, which is 68, divided by total number of recombinants, which is 1000, is 0.068. Likewise, between broad and cut, so broad and cut is 32 upon 1000, which is 0.032, and between angled and cut, it is 395 upon 1000, which is 0.395 recombinant frequency. So since the recombination frequency between B and C is the lowest here, which means that these two genes are closest to each other. So B and C will stay together, then there will be A. Sorry, here the B and C are close together. Now we have to decide the position of the C, as we can, A, as we can see that A and C recombination frequency is 0.395, which is more than the A and B, which is 0.68. So the A allele is closest to the B than to the C. So A will be here, then B, and then C, because A and C are farthest apart because of the most recombination frequency, which means that their distance is more between each other, which makes the order of the alleles as A, B, and C. Now, in order to calculate the map distance between the alleles, we can multiply the recombination frequency with 100. So for the map distance between A and B will be 6.8 centimorgans, B and C 3.2 centimorgans, and A and C 39.5 centimorgan. I hope you found the video useful. Thank you.